Welcome. Welcome everyone to session 4 of the FDA Science Forum on Product Development and Manufacturing. I'm pleased to introduce our first speaker, Jeff Ling. And with that, Jeff, take it away.
All right. Thanks so much, Suzanne. I hope everybody can hear me. So I'm just going to drive on. So yes, my name is Jeff Ling. I am the CEO of On Demand Pharmaceuticals. And we're going to talk to you about today is a technology that we've been developing for the past few years. By way of background, I should let you know that I was in the federal service for 30 years. I was an active duty U.S. Army colonel for 27 years. I served in both Iraq and Afghanistan. You'll come up in this talk. And I was the founding director of the Biological Technologies Office at DARPA, the Defense Advanced Research Projects Agency. And I spent a year also working for President Obama in the Office of Science Technology Policy. So I'm going to jump right into this talk. And so one would ask, what would an old, fat, ugly, retired Army colonel be doing developing new pharmaceutical manufacturing technology? And it really came from this. Back in 2003, shortly after 9/11, I was deployed to Afghanistan, where I was tasked to run the intensive care unit in the 452nd Combat Support Hospital, which you see in the background here. The person standing next to me is my friend Jim Eklund, who is a neurosurgeon. So we were sent to this place to basically provide care. During that time that I was there was a young American soldier that was brought in, and he was pretty badly beat up. And he needed a drug called bromocriptine for a condition called dysautonomia. And I really needed to get his dysautonomia under control because I wanted to send him back to Landstuhl, Germany, back to get more definitive care. Because there's only so much care you can deliver in a tent hospital like this. So, but unfortunately, I didn't have bromocriptine. So what we had to do was radio back to the airport, to Landstuhl, and they found a jet jockey who was able to get a box of bromocriptine and stick it in a flight suit and fly it to us. And when you think about it, it was really quite awesome that the Air Force came through and delivered this medicine that this young American needed. However, the cost of that box of bromocriptine must have been about $30 million just in gasoline alone for that jet jockey to fly his jet all the way over to deliver us this medicine. And so when I was there, I was thinking about this problem, and I said, geez, why in heaven's name don't I have bromocriptine? It's an old generic drug, it's cheap, so it's not a cost issue. And really the question became it was a logistics problem. And so, I said, gee, when you think about it, what is bromocriptine made out of? It's an organic compound. It's made of carbon, hydrogen, and oxygen, right? Just like most of the small molecule therapeutics, they're fundamentally organic compounds. So, yes, some have a little bit of, nitrogen in them. Some have some sulfur. Others might have a little bit of fluoride. But by and large, they're constitutively made out of carbon, hydrogen, oxygen, plus a couple of other things. So really what I'm saying is that you don't need the entire periodic table to make the small molecule drugs. You just need really a handful of agents. So in theory, you could, if you had a pencil and you had a glass of water, you basically have most of the constituents you need to go ahead and make most small molecules. Now, of course, we know the chemistry is not that easy, but conceptually you sort of understand this. And I should also point out, in addition to my MD, my medical degree, I also have a PhD in medicinal chemistry pharmacology that I picked up along the way after college, in between med school and college. I had to do something. So it was really that background in medicinal chemistry pharmacology that allowed me to sit down there and think about this problem in a different way. That, gee, if we had a machine with just some basic starting materials, we should be able to make a whole list of medications here in my little combat support hospital in the middle of nowhere. So, and that in fact gave rise to the program that ultimately that you'll hear about. And so after I came home from the war, I got assigned to DARPA, which was really kind of a transformative thing for me from a professional standpoint, but it let me go ahead and pursue this vision. So when I was at DARPA, I started this program called Battlefield Medicine. And the idea was to build a small manufacturing capability to make both small molecules as well as large molecules, i.e. protein-based therapeutics. The small molecule one is what I'm going to talk about today. And working with a couple of very brilliant scientists, we were actually able to kick this program off and go ahead and do this. And I would point out that when I started this program at DARPA, I actually had FDA officers come over and help me review the proposals and select the ultimate performers that we had on this project. So it was a real opportunity to work with FDA back when I was a government official. And so it was a lot of fun, and FDA has been awesome in terms of being part of this program since its inception, really. So, so the machine we called POD, or Pharmacy on Demand. And the three geniuses that really were the brains behind this was Klaus Jensen, who was chair of chemical engineering at MIT, Tim Jameson, who's chair of chemistry at MIT, and Alan Meyerson, who's a full professor of chemical engineering at MIT. And through their collective geniuses, they broke down the problem really into two main areas. One was the chemistry. Can you come up with chemistry, as I had talked about, where you're starting with more common materials and be able to make a whole potpourri of different medicines from different classes, right? So we're not cheating by making congeners of within the same class, but rather make different classes. And so that chemistry was part of it, but the other part was the machine, which was the engineering. So by bringing in both Klaus and Alan, having them work closely with Tim, you could see how this whole thing kind of came about. It's a hard problem, but it was a bounded problem. But if we were able to achieve this, and the way I pitched it to Dr. Tether, who was then the director of DARPA, to go ahead and invest the kind of money to go ahead and create this capability, which is what DARPA does, what would it do? I mean, it would provide a tremendous medical capability for our wounded service members, which you see in the upper left-hand corner, but it really opens an opportunity, a big space. It opens a space that you can now actually, if it's a little machine, let's pretend it runs off of solar power, you can actually put it in the developing world. And that folks in the developing world can now make the own medicines that they need. They make the medicines that they need, not be dependent on the medicines that they're given, but to actually make it themselves. Second, it empowers and liberates them. I mean, you truly empower somebody by teaching him to fish, not by giving them a fish, correct? And then on the lower left, we realized that if we could do this properly, we might be able to drop the cost of a lot of medications and, of course, take care of issues such as shortages and the like. So there's a lot of things that could come out from this little itty-bitty program that we had at DARPA. So we went ahead and executed this program, and it just marched along. And so we had to say, what would this machine do? I mean, that's what DARPA does, right? We go ahead and we say, it's not that you're going to build a machine, but what will the machine do? What would it provide? And it's really meant for our far-forward providers, for those that were in the combat support hospital like I was, or perhaps even further forward in the basic aid stations such as you would find in a combat outpost or forward operating base. So, number one, quality. Quality, quality, quality. It's absolutely essential that our service members get the highest quality medication. It's what they deserve, it's what they've earned, it's what we should be giving them. So in this process that I'm going to talk to you about, we bring up the notion of every dose quality, whereas every single dose, every single pill, every single injectable, you would know the quality of that, and it would be documented, and it'd be available for providers to look at, and potentially, of course, regulators. Second, flexible. This machine had to make more than one drug, obviously. Otherwise, what good is it? Three, resilient. It had to be able to operate under very austere environments. It had to run off of a military generator. It had to be able to take water from military purified sources. Potentially, it would have to run over solar, so on and so forth. It had to be very resilient because it is going into the battle space. Next, it had to be robust. These machines had to be able to be put on the back of a Black Hawk helicopter or into a Hummer. This cannot be something that's going to go on a semi, and it certainly can't be a factory. That was just not tenable for the kind of use case that we had in mind. It had to go on a submarine, it had to go on a destroyer, and it had to go in my tent hospital at the 452nd, right? So in essence, it had to be small enough to be man-portable and literally fit on these platforms that the military uses regularly. And next, sourcing. This cannot be a hat trick. The materials that go into the machine should be U.S. sourced, or close allied sourced, I should say. There can be no real dependency on potential foreign adversaries. So this really made the problem much harder, right? Because as we all know, many of the APIs that we're familiar with either come from foreign sources, and certainly the precursors for those APIs come from foreign sources. Not good enough for the U.S. military. If we're in a shooting war with some of these folks, we would be cut off from those raw materials, and we'd be out of luck. So the source materials really had to come further back in the synthetic chain for raw and fine chemicals that you can source either in the United States or very close allies. And finally, you got to make it cheaper or better. And it's as simple as that. I mean, that's it. So those were the criteria of what this machine ultimately would do when we set out to do it. That's the way DARPA does. We put forward criteria, and we expect the performance to meet those criteria. So what has come about is, in fact, the machine. The machine that you see right here that Brendan and Chris are wheeling is, in fact, the Pharmacy on Demand machine. And it has several sections to it, of course, but notably, if you see, may I have the pointer, please? Actually, I think I didn't ask for it, but may I have the pointer, please, Michael? And that, yes, thank you, is that you can see here, it actually has its own air handling system, so that it doesn't have to be necessarily hooked up to a snorkel or such thing. It's actually very small, but right here, this unit has what we call AMPOD, the American-made Precursors on Demand unit, a synthetic product, and what's not shown here is the formulator. But I want to be clear here. This is about starting with very raw chemicals, working your way through to key precursors, key starting materials, then to APIs, and then a final finished product. My soldiers are not going to do well with just APIs. They have to have a final finished product. So this was going all the way to final finished product, be it an oral or an injectable. ODP, the company that we spun out of the MIT event, is up and running. And right now I'm happy to say it's delivering. Today, if you go to Rockville, and you're all invited to come to Rockville, you can see that ODP is actually making medicine. So I want to be clear here. We're making key starting materials, we're making precursors, we're making API, and we are making medicine. All right? They're not FDA approved yet. We plan to file our first FDA submissions at the end of Q4 of this year because we're building on our GMP site. But I want to be clear here. We are making medicines today in the United States of America in Rockville, Maryland. The evolution of this program was actually fairly rapid. In 2011 was when I finally was able to get it kicked off because I was busy doing this prosthetic arm thing. And then and what happened was very rapidly the MIT guys built the very first prototype. So by 2013, they were actually able to make 7 different APIs. And then by 2016, they were able to make 14 different APIs. And very importantly, they demonstrated back-to-back manufacturing going from starting material through APIs through final formulated tablets in 5 different drugs. And they were able to do this in a campaign, and it's all published, by the way, over a 3-week period. They went back and forth between drugs, made thousands upon thousands of doses, and exceeded USP quality on all of them. So that was all done at MIT with the original machine. At 2016, we spun it out, and we started coming back and started to develop out the machine outside of the university system. And the idea there was being able to go and make other medications. We're up to 21 now APIs that the machines, this platform can make, and then also adding in sterile injectables to meet then the COVID crisis. So in 2020, of course, we all know the COVID crisis hit, and we were asked to scale up rapidly, and to actually focus on 3 critical ICU medications, one being propofol. And you see a photo of the propofol that's made on these machines, cisatracurium or Nimbec, and midazolam or Versed. And so these were 3 new drugs that we added to our armamentarium at that time, and that's what brought us up to 21 to where we are today. This is the kit system that you see here is to make it modular. And the idea for it to be modular so that you can switch it out if you want to make different classes of drugs, even though one class of drug can be made on a particular platform. So for example, you can make midazolam, it's a one-off to make chlordiazepoxide, it's a one-off to go ahead and make oxazepam, and of course it's a one-off to make diazepam. So we actually have these modularly built so that you can actually switch in and out of them. Will they always have to be modular like this? They probably not. We'll be able to find overlapping units, processes that can use the same material, but for the time being, they're modularly built out. You would say, okay, that's a pretty small machine that Brendan is standing for. How much can it make? Well, I can tell you right now that a single pod platform can meet the national demand of cisatracurium per day. This single platform that Brendan is standing in front of will make 2 million doses of Versed a day. This single platform that Brendan is standing in front of will make 40 million doses of fentanyl a day. So it just gives you a flavor of the manufacturing capability of this small yet mighty little machine. So you say, well, gee, Jeff, it's only good for the military. I will actually push back a little bit, and I would say, no, not necessarily. We now know from COVID-19 that we face drug shortages. We just know it. And the shortages come from a whole potpourri of reasons, and we won't go over them. You know them better than I do. But critically, we could say that if you had one of our little machines located in New York City, you could have actually met the needs that New York City would have had during that time for these kind of critical medicines that I just spoke to you about. The idea isn't that you're going to make the nation's supply of citracurium with one of those little machines. You just make enough that the New York City hospitals could use. Then you turn the machine into making fentanyl. And you make just enough that the hospitals need that day. And then you turn it into universal, and you get the picture. Because if you have one of those machines in New York City, and you have one in New Jersey, and you have one in San Francisco, the needs may be different for each of those places, but the machine by itself will go and handle it for that location. That furthermore, that's not only is it a highly flexible system to meet the needs of a local community, but also is highly resilient. If somebody came in and sabotaged that New York City machine, likely they can go to New Jersey and get another machine and get it to use. You can't take down a distributed network. That's the thing about the military. You know, it's always a distributed network. If you have a single hub, you're asking for trouble. And the military, it's a distributed system because that is how you build in resilience. I bring up COVID-19 not because it's an obvious thing to talk about, but because we're really not out of the woods yet. I think we all know that. The variants are still coming around, so the problem that we have already articulated may in fact rear its ugly head again. India has already been facing this problem, as you know, and they are a highly technologically sophisticated country. In fact, they make a vast majority of the APIs that we use in our country. So the fact that they are now troubled by this disease that's still raging in their country does lend itself for us to say that we have to understand that we still have to prepare for the future. And that's really what the point of that is. But in preparing for the future, why is it that the U.S. makes all of its own drugs? Why is India making the vast majority of APIs? Why is China making so many of our drugs and on it goes? Why is that? Really for many reasons, but not least of which is environmental issues, cost issues, and of course sourcing material. But I wanted to point out this little box on the right-hand side, and that is the comparison of wages. And this, by the way, I got actually from the FDA's database. And that is wages to make key starting materials and APIs in the United States. And we have it as a relative index of 100, which is the big bar in blue, which is the U.S. The relative labor costs to make the same amount of API in India and China are shown to you in the little bars on the right. And in fact, it's only 8% of the U.S. So in fact, if we build factories here in the United States, unless you want to go back to the 1890s where you're paying children 15 cents an hour to work in a factory, this is not a sustainable model here in modern America, and it shouldn't be either. The second thing is look where the source material comes. It sounds great that we're going to make API in the United States. That sounds just lovely. But take a look where the source material comes. Look at those flags. Those flags tell you that the precursors to make APIs still come from other countries. So if you don't want to include India or China, let's say in your supply chain for whatever reason, and you're going to make the APIs yourself, guess what? They've got you coming and going because you're going to have to get to them through the most common pathways using the most common precursors. You're going to have to go to China, you're going to have to go to India, you have to go to a number of other places. DOD can't tolerate that, but also in the world of the civilian world, if you wonder if this is a vulnerability that you really want the nation to have. And the disruption of the supply chain itself, irrespective of the markets themselves, is actually something we need to think about. COVID-19 has already shown us the fragility of the supply chain. And of course, and this is a photograph of the Navy in the South China Sea, that in fact, there's always going to be a threat to the potential supply chain. So we always have to keep that in the back of our minds. Because when you take a look at the right-hand pie graphs, they're pretty much telling you that the vast majority of the medicines that we use as citizens of the United States, 3/4 to 80% come from overseas. And then, of course, within the country itself, you'll notice that there are what we call pharmacy deserts. That is places where there are no pharmacy services available. And so one of our little machines in any of the clinics that are out there can, in fact, obviate this entire problem. The model that we see is one of our little itty-bitty machines, yes, it fits in the back of a Black Hawk helicopter, could just as easily fit in the back of an expedition SUV I mean, you could put one of those things in a rural clinic or a tribal clinic or anywhere else, and in fact, take care of their pharmacy needs right there. But the final thing that I want to talk about before I have to stop, and that is quality, quality, quality. We all know the issues of quality. I, as a practicing physician, and believe it or not, I still practice medicine. I still spend my time occasionally in the neuro IC at Johns Hopkins. You often wonder the quality of the medicine, how sure are we of it? I have patients who have epilepsy who will absolutely tell you that they're only going to want one brand of valproate, just one brand. They won't want any other, and it was Depakine. And why was it Depakine? Because if they took any other brand of generic valproate, they would in fact have a seizure. And I thought that was silly because, you know, valproate's valproate. And now looking back, and I'm wondering, we all assume valproate's valproate, but how sure are we? How sure are we, especially with batch processing? So in this situation, and of course this has been highlighted by Catherine Eben and others, and you guys know better than anybody else because you're the ones who take care of all of this. But what we have in on-demand pharmaceuticals, because the machines are standalone, we have it such that we have to measure at multiple steps of the synthetic pathway so that we know that we are in fact making what we plan to make. But that also allows us to measure quality at each step of the pathway. So for example, in our Cipro process, there are 65 separate steps going from precursor all the way to final formulated tablet, 65 steps. And even at the tabulated step, we have 5 separate monitor steps. This is, of course, to prevent subversion of the machine, but it's also ensure that the machine is making what it's supposed to make within specs. But lastly, very importantly, you now know the quality of each and every single dose. It's actually recorded, it's tabulated, and in many ways, I think that for full transparency, it should be available to FDA regulators, that they can go online at any time and look to see, oh, pill XYZ, what is its quality? And that is, in fact, something that we should all have. As a practicing physician, I would actually want that for every one of my patients, not just my military patients. That we should know the quality of every single dose. And the remarkable thing is, ODP did not invent the technology to do that. The online spectroscopy, the online monitoring, that's all off the shelf. Every drug manufacturer should be doing it. Every drug manufacturer should be doing it. If they truly care about patients, truly care about the health of a patient, you want to be sure that the medicine that you're putting in that person's body is in fact of the quality demanded by the the Food and Drug Administration. And so in my mind, this is the 21st century. This is something that we as a community of concerned citizens should demand of all manufacturers, just not ours. We're doing it because I believe that our soldiers deserve the very best. I was a soldier. I am a veteran. So, by the way, was Susie's husband. We served together. But at the end of the day, I believe in every American home. And ultimately, if we in fact put these machines out in these various communities, into the underserved communities, the lower socioeconomic communities that are existing within our own country, not just the headwaters of the Amazon or in East Africa or in the Philippines, but in our own communities, we're now reducing barriers of health disparity, that we're making available medicine. And in fact, by our calculations, with the medicine that we are making, we can cut the cost of medicines by at least 50%. And there's a lot of reasons for that we'll go over another time, but we can actually reduce the cost of medications for our patients. There's no reason in my mind that grandma should have to forego a meal because she needs to buy her medicine. That is just fundamentally wrong. And when you look at manufacturing, sure, the prices can come down. That savings has to be passed on to grandma. There's no reason why it's not. And again, we can now start servicing areas of the world that simply don't have their own medicines. When you give things to people, you put them in a welfare mind. When you teach them how to make for themselves, you give them empowerment. That's what this is all about. Giving them a machine that can make what they want, when they want it, how much they want it. That, in my mind, is really what's going to make an impact and help all around. And what do people need? I served in Afghanistan. I went over there 5 times. I did 5 different deployments over there. And I will tell you what they want. They want clean water. They want education for their kids. They want a safe community, and they want good health. These are the fundamental rights of every human being. And yet, giving them clean water is something we would do by drilling deep wells. That's very straightforward. We can do that. Giving them good health, let's give them the medicines that they need. They don't need monoclonal antibodies. They don't need CAR T therapy. They can do just fine, thank you very much, with a couple of the anti-infectives that we all know and well that have been around since the 1940s. They can do just very well, thank you very much, with a little bit of cisplatin or a little methotrexate. We treat a lot of neoplasms that thing. And you can treat a lot of diseases with some very simple medicines. You do not have to give them CAR T therapy. The access to inexpensive, high-quality medicines that they need will then be absolutely transformative as a country. Think of the things that they can achieve with that. So this is our pharmacy on-demand system. You can take a look at it. It's in the upper right-hand corner. We actually make all the parts ourselves in our machine shop, which is the middle panel, and we're building out our GMP suite so that we can actually do all the things necessary and critically important, I might add, that FDA will require of us before that we can be FDA approved to go ahead and get our medicines out into the clinic. Because at the end of the day, this is not a science fair project. This is actually a capability project. It is so we can make medicines, and which we're doing right now, but make them to the quality and assurance that the FDA will allow us then to get it out to the clinics. My soldiers need these medicines, and I want to get it to them. But you can see the small size and all the capabilities around it. I don't have to tell you guys what to do because you know how to think about this problem. You do it all the time. So in the end, I see there's a partnership. There's a partnership with FDA. I believe that what we can offer to the FDA is, in fact, an every dose monitoring system, every dose monitoring system that, in fact, can be the standard of the industry and something that we can ask of all manufacturers so as to protect our own citizens. We actually have new mechanisms to foster innovation. This small platform allows us to actually make orphan drugs at an economically feasible way. We don't have to sit there and charge them these exorbitant rates to recoup manufacturing costs. Our little machine can go ahead and make many of the small molecules that are necessary. And second, it allows us to open up the opportunity to think about what else is possible. What else can we do to create a new future? This is the 21st century. To be dependent upon offshoots of 18th century England's Industrial Revolution approach with smoke burning coal stacks and child labor is just ridiculous in my mind. We have serious problems today. We can all meet them together. But it does require a partnership between the government and industry as well as what was and what will be. So I think together we can embrace what will promise to be a brand new future. So thank you very much, Susie. I yield the floor back to you.
Thank you so much, Jeff. This is really a great presentation. And I don't know, do we have time for any questions in the chat, or no, just go on to the next one? Okay, you're gone. Okay, thank you. And our next speaker is Elizabeth Jones. Hi, Elizabeth.
So, thank you for the introduction. So today I'm going to tell you about how we've been using multi-imaging mass spectrometry in toxicological studies. So first of all, I'm coming to you from the National Center for Toxicological Research. We're actually a branch of the FDA that's located in Jefferson, Arkansas. So I work within the Division of Systems Biology, where our mission is to use system biology approaches to address problems of food, drug, and medical product safety. Also falling within this category is our ability to innovate, to evaluate innovative technology, which multi-IMS falls under. So essentially what we're doing here is trying to address the emerging health threats and regulatory research needs and fill in any knowledge gaps where we can. So MALDI stands for Matrix-Assisted Laser Desorption Ionization Imaging Mass Spectrometry. It was originally developed in the 90s by Dr. Caprioli from Vanderbilt. The main advantage of this technology is it allows for the detection of an analyte of interest across a tissue section. It's very robust in that we can analyze a tissue section and see hundreds of analytes within a single imaging run, and it's also label-free. So the way this method works, starting with a mouse brain here, we can take tissue sections from this organ, which we put on a conductive slide. We add an energy-absorbing matrix, and then we put this into the mass spectrometer. Now, we're very fortunate at the FDA to have a Bruker SciMax 7 Tesla FTICR, so this really gives us very good sensitivity and extreme resolution. So once the slide is in the mass spectrometer, it'll use a laser and it'll fire across the entire tissue section. At each tissue section, at each place the laser fires, it's going to generate a mass spectrum, such as this one seen here. After it rasters across the entire tissue section, you'll get an overall mass spectrum, which then we can go back in and look at specific peaks. So for example, we could look at this peak right here at this mass of 736.5. and I can show you in this first panel here the distribution of that peak across this tissue. We're looking at this as a color scale, so areas of red would be where this is higher and areas of blue would be where it's lower. And of course, we're always looking at this in relation to the tissue pathology. So I can show you one single analytes here, which I've done for both of these, and we can also show you overlays. So now what I want to do is take you through some projects that we're utilizing MALDI imaging within the FDA. The first project will be a cardiotoxicity study, and then clearly the opioids are a huge focus for us at the FDA, so 2 opioid studies. And then I'll end with a very recent project in which we are looking at N-link glycans in some COVID-19 tissues. Okay, so the first study here, and this is led by Dr. Tao Han, and he's interested in looking at identifying early biomarkers of sunitinib-induced cardiotoxicity. Sunitinib is a cancer drug. It's been mainly used to treat renal cell carcinoma. However, it does have this off-target effect of being cardiotoxic. So Dr. Han is using a pretty broad omics approach to look for these biomarkers, but he approached the MALDI imaging team to see if we could see sunitinib distribution across his tissues which are dosed. This is just a pilot study. Here's his drug concentration. So we picked the highest dose at 120 mg per kg. Obviously, we're looking for sunitinib, so here's the parent molecule here. He delivered the apex of the heart tissues for us, which is at the tip of the heart here. So we took these heart tissues, we sectioned them at 10 microns. We used DHB as a matrix and a 50 micron raster width. If you come down here and look at the MALDI imaging data, you can see that we were able to detect the sunitinib, and it does seem to maybe co-localize to specific areas, which we were following up on. And sometimes it's interesting to overlay the drug distributions with the blood, and we know we're in the heart, so there should be blood there. And we can see that the tissue distribution of the sunitinib is not tracking exactly with the blood, but it seems to be in tissue areas. So this is just a pilot study, and we will increase the numbers in this study and hopefully collect full hearts for the next analysis. So this is one of our opioid studies, and we're doing this in collaboration with Dr. David Thorne. He is interested in visualizing opioids and opioid metabolite distributions in the hopes that it could offer insight into addiction and metabolism of these drugs. So in the pilot study, what we wanted to do is make sure that we could detect these 2 opioids he's interested in using MALDI imaging. And he's interested in oxycodone, shown here, and then morphine, I'll show you on the other slide. So with this study, he dosed the animal at 5 mg per kg. We collected the rat brains, which we sectioned at 10 microns, used DHB as a matrix, and then we have 100 micron raster width. Now, we did the first initial run of this tissue. We did not detect oxycodone. Fortunately for us, the instrument has a feature called KZ, which we can tell it only select this specific ion. We went back in and used that method. You can see here we could detect oxycodone, and we were pretty happy about our level of sensitivity, as you can see in relation to this curve. So morphine we did in the same way. The dosing was a little higher, so we were at 20 mg per kg here, but we collected the brains and analyzed it the same way. Unfortunately, morphine was not as straightforward. We could not detect the parent morphine. However, now one of the advantages of MALDI imaging is you can see drug metabolites as well in the same imaging run. So we could detect the 2 major metabolites of morphine. However, they do have the same mass, so when I'm showing you this picture, it's just an overlay of both. So we're working on the sensitivity of this method, and hopefully we can detect the parent morphine in future studies. So here's another one of our opioid projects. This is led by Dr. Amy Inselman, and she's actually giving a talk tomorrow, so I encourage you to check that out if you want to see more information. But a little background. In 2015, the FDA identified a possible link of opioid exposure in early pregnancy and an increase in neural tube defects. At this time, they didn't feel like there was enough information to change any indications of the way it was being prescribed. So since this time, we've added a lot of studies to try to address these gaps. So in this study, she's taking maternal mice at gestational day 8 and exposing them to various opioids, and then they're sacrificing them at day 18, and by C-section, we're collecting the mouse pups and fetuses to look for neural tube defects. So I won't go much into these except just to point out that one of the main characteristics of neural tube defects is an abnormal development of the brain. So you can see when she's using her opioids here or exposing the maternal the mice, that she is actually recapitulating that in the mice. So for MALDI imaging, our goals, we had 2 goals here initially. So we wanted to see if we could see any residual opioid hanging out in the fetus, and we also wanted to see if we could see interesting lipids or other markers which may track with neurotube defects. So we just picked, here's a representative of the two tissues we used. Here's a normal fetus, and you can tell the brain here is pretty normal. We picked a fetus that was dosed with a high level of methadone. You can see here the brain is not developed. So with the imaging, we sectioned it 10 microns. Again, we used DHB, and we ran this with a 100 micron raster. So here's some of our preliminary data. We could not see the drug. However, we weren't very discouraged by this since the last dosing was 10 days prior to the SAC. But we did find some interesting sphingolipids, and so you can see up here is the normal fetus, and down here is the dosed fetus. These sphingolipids that we found are known to be involved in neural tube development, so we were very interested and excited to see this. And you can see sphingosine 1-phosphate and ceramide. We actually can see a lot of the ceramides, and you can see that it's elevated in this area right here in the normal fetus versus the abnormal, and you can see it's in this area where there's extraneural tissue. So this will be something really interesting to follow. We have many more tissues to analyze, and we will keep an eye on that as we go. So one thing we want to add to all of these opioid studies, and this is a very recent application that came out of Dr. Per Ongen's lab, in which he's developed these reactive matrices, which allow for us to now do neurotransmitter detection. So we feel like, just showing some examples here, so we really feel like we're going to add this in very soon, and this will really help add a new dimension to our opioid imaging studies. So lastly, I want to talk to you about a project in which we're working to characterize the effects of viral load and immune cell infiltration in autopsy tissues. And these are taken from COVID-19 patients, and these are lung tissues. So we're doing this in collaboration with Dr. Richard Drake and Dr. Peggy Angel and Dr. Anand Mehta, and we'll show him here in this picture. But they're at the Medical University of South Carolina, and we're also getting our tissues from Emory. So essentially we want to characterize the microenvironment of these virus-infected cells and immune cells. And we're actually using two different imaging modalities, but I will only focus on multi-imaging here. So we're interested in actually looking at the N-link glycans in these COVID-19 positive tissues. And of course, we want to correlate these back with all the immunohistochemistry in an effort that we could potentially identify biomarkers or targets for therapies and vaccines. So you may ask why N-link glycans, but if you look at the surface of the SARS-CoV-2 spike glycoprotein, you'll see that it's highly decorated with these glycans. And so when we're talking about glycans and glycosylation, what we are referring to is the covalent attachment of a carbohydrate or glycan to a protein or lipid. These can be N or O linked. It's the most common post-translational modification, and it mediates many activities. Therefore, when this goes awry or you have aberrant glycosylation, you can see impaired immune function, and we see this in a variety of diseases, including cancers. So MALDI imaging offers us a unique opportunity to look at this N-link glycan distribution across the tissue section. So here I'm just showing you the structure of these glycans. This is the core of an N-link glycan. They can be quite simple, and we can get quite complex as we add in more sugars. The ability to look at this using imaging is really made possible by this enzyme known as Pingase F. Pingase F has been around for a while. However, it's been re-engineered by our colleague, Dr. Anand Mehta, and he's made it more stable and much more efficient. So it cleaves actually here, this asparagine, and it releases the N-link glycans. So for the sample prep here, it's a little different on the front end. Now we're using clinical tissues, so they're fixed in formalin. So we have a step here to get rid of the paraffin and reverse the antigen retrieval. However, once we do that, we can incubate with our enzyme and then go on with our matrix application and MALDI imaging as we've shown previously. So I'm just going to talk about trends here in this with these tissues because the study is ongoing and we have many more coming, and we're not far enough down the road to make true conclusions, but we'll show you and share with you some trends. So the way we're assessing these trends, and you can see the control tissue versus the COVID-19 tissue here, we can generate pictures such as these where I can tell you every single image is a different glycan or a different glycan distribution. So we can really start to compare these different glycans between the groups, control and COVID-19. This is kind of how we're tracking trends. We also can, of course, overlay multiple analytes to see where they are in relation to one another. So one of the trends that we've seen thus far that seems to be pretty interesting is we're seeing these high mannose glycans, and they're seeming to track with a T-cell marker. So over here you have your COVID-19 positive tissue. We can expand out and look at the H&E and then the CD8 staining. The CD8 is a T-cell marker, so CD8 staining would indicate T-cells. And so we see in these areas of high CD8 staining, we see these high mannose glycans. So maybe a preliminary hypothesis would be that these indicate acute phase and inflammatory response. However, as I mentioned, the study is ongoing. Another trend that we've seen is a decrease in sulfated glycans in the COVID-19 tissues. So we know sulfated glycans are anti-inflammatory, so this could make sense, and again, something that we're following. Now, switching gears just a bit, we're also very interested in assessing the extracellular matrix, and we know that collagen is very important within the extracellular matrix, and there's a lot of tissue remodeling that's occurring on the surface, and it's part of fibrosis, and we're actually seeing some of this fibrosis in the COVID-19 patient tissues. So actually, Dr. Petty Angel created this method where now we're switching to a bacterial collagenase instead of the pangase F to release the collagens, and so it releases the collagens on the surface, and I'm just showing you a handful here, but essentially we will track this moving forward as well, so we can get an idea of what's happening with this tissue remodeling and with progression of COVID-19. So lastly, and I think this is one of the coolest slides here, and it's some of the most recent data that we have coming out of Dr. Drake's lab. Again, this is a method that was developed out of his lab. And so in this slide, we're back to glycans, but we're showing you some sialylated glycans. So sialylated glycans, and here's the structures here, they're negatively charged glycans. They can have various linkages, so for instance, a 2,6 linkage versus a 2,3. So this is a very recent method in which they can use chemical derivatization to actually see the difference between an alpha-2,6 linkage and an alpha-2,3 linkage. So interestingly, I mean, in the viral world, we've heard of respiratory viruses that can use alpha-2,3 and alpha-2,6 for viral-mediated cell entry, and even some of these respiratory viruses may prefer a different linkage over the other. So we know with COVID-19 that it binds the ACE2 receptor. However, there's been emerging evidence that suggests that the sialic acids may also play a part in this for the viral entry into the cell. So in these images, he's actually showing, we're seeing an increase in these sialic acids in the COVID-19 tissues. And you can see here, interestingly, obviously it's a different distribution. And so you can tell it's different, the different linkage. So in conclusion, I think we've shown the incorporation of multi-imaging workflows has proven useful across a diverse range of FDA projects. We're getting new projects daily, which is exciting. Our future opioid brain studies, we will expand and also add in the neurotransmitter component. Now, we've only had our instrument up and running for maybe a year, but we've had some setbacks with COVID, as I know everybody has, but we really haven't pushed our instrument as far as resolution goes, and we're really looking forward to doing that in the future. Also, we know that multimodal imaging and or incorporating different imaging platforms or existing ones with multi-IMS is a very important area. And so we're really actually trying to do this with our COVID-19 work, And so we think this will be a major theme and area of expansion for the FDA and the multi-IMS group. So with my acknowledgments, I want to thank a lot of members of the DSB, including Richard Beeger, Laura Schnackenberg, Tal Hahn with Assist Tissues, David, Amy, and Jessica, for our pathology folks who are very helpful with annotating our tissue. Of course, the Medical University of South Carolina, and I did not mention that James Dressman ran all the images that you saw, and then also a couple guys at Bruker that have helped us going, and Emory for the autopsy tissues. And thank you for your attention.
So thank you very much, Elizabeth, for some really exciting research and presenting it today. And we look forward to hearing from you more at the next Science Forum. So thank you. And our next speaker now is Sabina Seitz. Sabina, you're up.
Thank you. Thank you, everybody, for attending today's session. Oh, that is not my presentation. Okay. FDA aims to identify and develop evolving and innovative technologies and tools to support alternative methods to traditional toxicity testing. These tools may help bring FDA-regulated products to market faster, prevent products with increased toxicological risk from reaching the market, and to prioritize chemicals on the market for reassessment. Importantly, these tools may help replace, reduce, or refine testing. One such tool that I'm going to talk to you today about is the expanded decision tree. So what is the expanded decision tree, or EDT? It is a sequence of chemical structure-based yes or no questions that either leads to another question or the assignment of the substance to one of 6 classes of relative, chronic, or toxicity. The schema on the right does not really do justice to the EDT. While you can see the 47 main questions, what this drawing doesn't show you are the over 100 sub-questions and the numerous sub-questions that really help us refine the classification of compounds according to their toxic potential. DDT can be used with all chemicals except elements and inorganic substances, proteins, and unhydrolyzable polymers. Currently, they are out of the applicability domain of the EDT. The basis of the EDT was the 1978 Kramer et al. Decision tree that was not updated for over 4 decades. In addition, we used a collection of toxicological, absorption, and metabolism data and informed structure-activity relationships on thousands of compounds. As I briefly mentioned before, the EDT classifies compounds into 6 different classes of toxic potential. These range from class 1, non-toxic, all the way to class 6, the class of extreme toxicity. Just a second, I lost connection. Okay, I am back, yes. Apologies, for some reason it seems I lost connection for a minute or so. Each EDT class is associated with a so-called TTC, or threshold of toxicological concern level. As long as the intake of a substance does not exceed its class TTC level, we assume that the intake of the substance at that level is safe. Combined with the TTC levels, the EDT can be used for safety evaluation. It's a tool in our toolbox. And unlike many other tools, we can predict chronic toxic potential and not just acute toxicity. The EDT can also be used for screening and prioritization of chemicals for further testing and to identify compounds of concern. It can be also used for read-across. Some of you may not be familiar with this term. Sometimes we have a problem that we need to evaluate the safety of a compound for which either we have no toxicological data or we don't have enough toxicological data. So what do? We try to find a data-rich substance that is structurally and metabolically, hence toxicologically similar, and we use the data from the data-rich substance to help us evaluate the safety of the data-poor substance. Now you may ask, how can the EDT help with this task? 2 different ways. We designed the EDT questions in a way that most questions, sub-questions, and sub-sub-questions classify compounds there that are structurally and metabolically similar, hence can act for each other as read-across substances. So if you have a data-rich substance and it gets classified as the same question, you can be confident that you can use the data-rich substance as a read-across substance for your data-poor substance. Also, once our software is completed, we are going to connect the software to our large toxicological database that contains subchronic and chronic toxicological data. And the software will help identify the user read across substances for their data poor substances. The EDT is also suitable for mixture evaluation. And we have a procedure that takes into account synergistic and additive toxicities. What do I mean by that? Sometimes components of mixtures can be related structurally and metabolically, and their toxicities can be increased as a result. And our procedure is capable of detecting to ensure that we account for that. We also have a procedure to determine the sufficient similarity of mixtures. What do I mean by sufficient similarity? Sometimes, for example, in the case of botanical extract, depending on the growth area, the soil, the amount of sunshine or rain the plant receives, how you process the plant, and how you manufacture the extract, the composition can vary. And you cannot test every different composition. So if you, let's say, test one, how do you know that the others other mixtures are sufficiently similar that you can use the tox data from one to evaluate the safety of the others? So we also developed a procedure that can do that for you using the expanded decision tree. You can also use the EDT for post-market to ensure that compounds at ever-changing intake levels, oftentimes increasing levels over time, are still safe. So what's the status of the project? We have completed our first two papers. The concept paper tells you what we did, why we did, and how we did it. And the main article contains the EDT, a little bit of explanation after each question, what really the questions mean, what's the basis of the question. And at the end, we have a large training set, a large number of compounds for which we show how they traverse through the EDT. We did that to help people get comfortable using the EDT manually until our software is finished. These two articles are currently undergoing FDA clearance. I'm hoping to be able to submit both of them to a journal for publication, and they are going to appear side by side in the same paper, in the same issue. We are also waist deep in the external validation of the EDT. With the help of EPA, we have created a second large database of compounds that we do not have in our current EDT database. And we have subchronic and chronic toxicological data for all of these. And we are using this to validate the EDT to show that it's fit for its purpose. That said, based on the current results, we are not expecting any surprises, and we have already started writing up some of our results. And we hope to finish the external validation and the associated publication by the end of this year. As I told you, we have a procedure for mixtures and the evaluation of the sufficient similarity of mixtures. We simply need to write them up. We should be able to do that sometime the beginning of next year, which is going to be our fourth publication. In the future, we would also like to publish a series of articles that provide the users with the detailed scientific basis of every single EDT question. In addition, we want to use these papers to kind of give an overview of the structural activity relationships for the whole chemical space out there. So these papers could be a resource for the scientific community and not just for those that use the EDT. As a result of this project, we are going to have two large databases with thousands of compounds with subchronic and chronic toxicological data that will serve as a resource for FDA. In addition, these databases will be connected to our software to help you provide data-rich read-across substance for your data-poor substances, to help you evaluate the safety of your data-poor substances. Unfortunately, we ran out of funding for the EDT software, so we stopped working on the software last fall. We are currently in the process of trying to secure more funding so we can resume work on the software. In the future, I would like to regularly update the EDT. As I mentioned to you, one of the bases of the EDT was the 1978 Kramer Decision Tree, and it was not updated since 1978. And that's something I do not want to happen to the EDT. So once we have published our first 2 papers, I would like to call upon the scientific community to give us suggestions on how we might improve certain questions and to provide us with the scientific basis for the change that's recommended. And I am going to take a look at every single recommendations, and as long as the data supports it, we will make sure to incorporate it into the EDT questions. And every few years, we would like to publish the updated EDT to ensure that it's always up to the current state of science. That said, we are actually planning to design the EDT software in a way that would enable us to relatively easily edit the questions within the software. So it wouldn't take a lot of time and a lot of resources to update the software every time we update the EDT. So what are the goals of this project? To provide FDA and the scientific community with a fast and efficient tool capable of predicting the chronic oral toxic potential of both individual chemicals and mixtures, and to provide safe intake levels for them. This tool can be used for safety evaluation and screening and prioritization for further evaluation and testing. It provides scientists with a tool that can help replace, reduce, or refine animal testing, and to advance our understanding of structure toxicity relationships. Our team is small, and we work on numerous other projects concurrently. But thanks to the dedication of the team, the first 2 papers are undergoing clearance right now. I would also like to thank CIFSEN and the Office of Food Additive Safety leadership and scientists, and all those who have reviewed the EDT publication. Their suggestions helped us greatly improve the first 2 papers. I would also like to thank Drs. Anthony Williams and Richard Judson from the US EPA for their help with creating the external validation set. And thank you all for listening.
Thank you very much, Sabina, for a very interesting talk. And next up is Chris Lustig.
Excellent. Thank you very much. I'll wait for the slides to appear. There we go. So unlike a specific research project, what I'm excited to tell you about today is a new pilot program that exists within CDER, specifically in the Office of New Drugs, that's embedded as part of the biomarker qualification program. Ultimately, what we're trying to do is bring forward innovation in drug development as a tool. But what we found over the many years, even though we had existing programs for different tool types, namely biomarkers, clinical outcome assessments, and animal models to be used under the animal rule, that there are many different novel technologies beyond those 3, and they just didn't have a home. And so when they would get proposed in an individual IND, or it would come up to a member of the community, it would kind of get bounced around either within an office or even between centers. And so as part of ISTAN, which stands for Innovative Science and Technology Approaches for New Drugs, we now have a pilot program for these innovative tool developers to sort of have an interface by which those tools could be proposed and evaluated by the FDA staff. So currently the qualification programs are functioning under the 21st Century Cures legislation, which was enacted at the end of 2016. And in that legislation, they define drug development tools as any method, material, or measure that the Secretary determines aids drug development or regulatory review. As part of the legislation, it did call out and define both biomarkers and COAs, but it also gave us the authority to bring in other tool types that currently do not fit into either of those two buckets, if you will. And so the ISTAN pilot is listed at the bottom right, is a pilot to bring in these novel technologies for evaluation so that we can have conversations around them and their potential use in aiding drug development. So what am I talking about around novel technologies? So I just wanted to give you a flavor with some examples that are listed here in the slide. So certainly there's a large interest in pharmaceutical clinical trial oversight to enable them to be either remote or decentralized. There are many advantages to that, and that enables more people to potentially participate in clinical trials. But that remote or decentralized setting sometimes poses some challenges for us, at least in the drug world, of how we collect data and how the tools that are used in the clinical trial are evaluated. So for instance, in some clinical trials, there are clinical reported outcome measures called ClinROs, where they may be collecting information through telemedicine interviews. And so there may be a need to standardize that process to make sure that the data that's collected is beneficial to us as part of that individual drug development effort. Likewise, also in this decentralized space for clinical trials that are looking at drugs to aid in treatment of dermatologic conditions, rather than doing an in-person physical exam of the skin, there are multiple groups that are wanting to do digital photographs of the skin that can then be sent into a central reader. And there's challenges associated with that, but it certainly shows a lot of promise. Likewise, we know that there may be tools that can advance our understanding of drugs, especially around safety or toxicology concerns. And so there are various tissue chips or microphysiologic systems or in vitro systems that have been proposed to potentially aid in the normal studies that we receive from the farm tox data in the animals. And so there's an interest in bringing forward these novel technologies for many different reasons to provide supplemental information. And then also in digital health, there's the use of artificial intelligence and algorithms for machine learning to potentially look at data across to look at novel endpoints and inform study design. And then finally, there's also an interest in for the medications that are administered in a clinical trial to have some way of tracking whether or not a patient has actually taken the study drug. And so there's various adherence sensors that, again, folks have proposed. So this is just a little bit of an overview of the type of novel tools that we're speaking of that certainly, if found to be beneficial, could really aid in drug development. And so ISTAN is intended to provide a mechanism whereby we can have conversations around these novel tool ideas. What we're trying to do, in addition, by centralizing this under a banner program, is that rather having individual conversations happen within INDs and within sort of siloed groups within FDA, that by centralizing the conversation, that also allows us to hopefully aid our staff by bringing to their attention what tools are being brought forward in other divisions or even in other centers, so that we can sort of have a shared communal knowledge, and then also bring forward hopefully what will be standardized recommendations so that the outside parties are going to be hearing a sort of unified FDA voice rather than multiple groups, perhaps differing opinions. And so by centralizing it, it does allow us to bring forward that sort of standardized advice. It also increases our knowledge around these technologies more generally. And then by also having a staff with an iStand also reduces the workload and the expectations for individual groups because we can help to simplify and augment the conversations. So hopefully that will speed in the overall decision-making process. Now, iStand is embedded as a drug development tool in a pilot format. And certainly qualification is one of the things that we would hope to be achieved as part of this engagement. For those that aren't familiar, qualification basically gives a regulatory endorsement for the use of a tool independent of a specific drug program. And the use of that tool is defined by what's referred to as a context of use, which kind of clearly delineates how the tool was used and how the data that was brought forward was interpreted, so that in the future, if another pharmaceutical company wants to use the same tool for the same purpose, there's no need to bring forward the data over and over again on their part, and on our part, not the need for re-review each time. And so there's a real advantage to this qualification process. At the same time, we also understand that the novelty of some of these proposals is really kind of rather complex. And so there's going to be a lot of taking the proposals and ideas and breaking it into constituent parts that are manageable to have conversations around them. And for some of the technologies, there may be so novel, there may not even be precedent within the FDA community. And so some of those outcomes may not be feasible as a qualification effort, but rather that what we could do is have public meetings, create white papers, or even guidance around best practices. So within iStand, once a submission comes in, it gets internally triaged. It gets evaluated both by the staff on iStand itself, gets presented before a center-wide committee called the Drug Development Tool Committee for additional inputs. And then ultimately those that are prioritized can come in, and then based on the tool, the novelty of the technology, and the data that's available to us, it will either undergo the qualification route as shown here on the top, or perhaps it will get shunted to a public meeting, a white paper, or guidance. So some of the key features of the pilot is that we certainly understand that FDA staff are being pulled in many different directions simultaneously. And the last thing we want to do is just to add something additional to their plate. So while there is value in these proposals, we also understand that until and if this is going to be appropriately resourced, we do have to limit the number of submissions that can be entertained for the pilot. And so at present, we're thinking 2 to 4 per year. And they're going to be triaged based on public health impact and the availability of FDA expertise. The pilot itself went live back in November, and since then we've had many conversations with outside developers around many different ideas that they've put forward, and the level of interest is very high. There's also going to be a learning curve internally and externally, and so I think over the next number of months and during this initial year of the pilot, we're going to certainly continue to make adjustments to the pilot to make sure that the resources are available publicly, as well as the information that is considered for the pilot, but ultimately we're going to have some really potentially high impact projects in the pilot. Certainly there's cross-center interest in these tools, especially between the 2 drug centers, namely CDER and CBER. And so the role of the Drug Development Tool Committee I alluded to earlier is going to be really instrumental in providing that sort of senior level oversight and expertise to really truly evaluate the potential impact of these tools. It's going to be the same regulatory framework that we use for other drug development tools, namely COAs and biomarkers. And so we're using existing tools, processes, and best practices for iStand, so we're not reinventing the wheel, so to speak. And then the iStand staff are going to help to coordinate the conversations with the external groups. They're going to help to identify internal experts and facilitate those conversations, aid in the storage of the information as part of ongoing knowledge management efforts, and then also coordinate with existing programs to ensure that whatever project proposal that's brought forward has its best home, since there's many different groups across FDA centers that are looking at this novel technology space. I draw your attention that there is an iSTAND website that's live. On that, you will find information around the content that we expect to see and the submissions that come forward, as well as just some general information around what would fall within scope for iSTAND and what would not. So I appreciate the time today to bring this to your attention. There is an iSTAND mailbox that's listed here on the slide, and Steve Berman and my group is the lead for this particular project. So if you have any questions, please feel free to reach out either to the mailbox or to Steve, and we'd be happy to engage with you. Thank you very much.
Thank you, Chris. Just a couple of minutes, I'll ask you, have you started working on any submissions, or you're mostly in the consultation period right now? Or? It's a very exciting program. It's gotten a lot of really good feedback.
No, absolutely. Most of our conversations so far have been in the microphysiologic system in vitro system space. We've had, I think, more than a dozen letters of intent that have been submitted that we've had conversations with so far. The kind of challenge, and this is what I alluded to in the presentation around the learning, both internal and external, is that a number of these systems that have been brought forward around screening compounds and such. So the context of use is not an area that FDA regulates. That doesn't mean that it can't aid in drug development. It's just out of scope for the program. That said, we have been working with some of those developers to see if we can identify an area where they could be beneficial, especially around safety and toxicology assessment. And so we've been having back and forth, and hopefully we're keeping our fingers crossed that we're going to get a proposal coming in that space that is for a context of use that our staff think would be really beneficial if successful.
Well, thank you very much again for presenting this exciting program at the Science Forum. And
thank you for the invite.
With that, we'll move on to Kevin Fu.
All right. Great. Usually we don't say we get started with cybersecurity too early, but today we will. Hello, my name is Kevin Fu. I'm the Acting Director for Medical Device Cybersecurity in FDA's Center for Devices and Radiological Health, and also Program Director for Cybersecurity at the DHCOE. Just a moment as I get my browser set up. All right, I'm not going to read this bio, but my background is in research in medical device security. I'm probably most known for some of my early work on discovering vulnerabilities in pacemakers about 10 or 15 years ago, showing how a homemade transmitter can cause the conditions that would lead to a fatal heart rhythm wirelessly, in particular ventricular fibrillation. And so that really caused a sea change in how to design medical devices to resist computer security threats. So I've been talking about this for quite a while as an outsider, as a professor in academia and engineering, and today I'm wearing the hat as a representative of FDA, and I'll give you a perspective from FDA on medical device security and how to make sure that they remain safe and effective despite inevitable computer security risks. So let me just share one fact, and that is FDA has elected not to approve and not to clear medical device submissions based on cybersecurity concerns alone. So let that be known. They are significant risks that are expected during the pre-market review process. And why? The reason why, simply stated, is because cybersecurity is safety. It's extremely difficult to have a safe and effective medical device if there are cybersecurity risks of clinical relevance. So to try to get out in front of these risks, FDA has invested a huge amount of effort over the last decade on providing guidance to industry on FDA expectations for built-in cybersecurity rather than bolted on after the fact. There are two primary guidance documents available. A pre-market guidance document finalized in 2004. It's essentially the engineering that the FDA review staff will look for when evaluating the pre-market submission of a medical device. And a second document finalized in 2016 is called the post-market cybersecurity document. And that I would consider more of a social sort of engagement document. It talks about how medical device manufacturers and health delivery organizations can work together to share information about not only vulnerabilities in a medical device, but actual incidents when an incident occurs, such as ransomware affecting a medical device, and how to communicate that with different stakeholders across the medical device ecosystem. There is also a revised version of the pre-market guidance that was not finalized, issued in 2018, and we have an upcoming version of the draft pre-market guidance targeted for public comment beginning in late 2021. Let me give you a couple other highlights about FDA's activities in protecting medical device cybersecurity. Number one is threat modeling. It is the cousin to hazard analysis. The idea is that it's very difficult to make scientific claims about medical device security if a manufacturer doesn't provide a reasonable and refutable threat model specific to the medical device. So FDA stood up a sequence of boot camps for manufacturers through the MDIC and MITRE to really train the trainers and teach individuals how to create more effective threat models for medical devices. And I'll tell you a little bit about threat models in just a moment. Another big area that FDA is working on is something called SBOM, and software bill of materials, and that's actually through the International Medical Device Regulators Forum. The reason for working through the IMDRF is to help harmonize different standards and guidance across the borders of countries. This is all about how to get a total product life cycle to include cybersecurity and to bring more consistency and harmony across the borders for more certainty for the manufacturing community. Now, SBOM is an acronym, therefore it's a real thing, and you're likely going to be hearing much more about SBOM, not only in the FDA medical device space, but in federal systems all over the country because of the recent executive order from President Biden, which specifically mentions SBOM inside the executive order. So a software bill of materials is effectively a nutrition list. It's sort of an ingredient list of what third-party software is on the inside, in this case, on the inside of a medical device, because medical devices are not too different from any other kind of operational technology, OT system, in that it consists of a large number of components, many third-party components, both mechanical and software. This may include things such as operating systems, various libraries of software, but also it can include firmware and other components deeply embedded both on inside of the medical device and even on the outside of the medical device, such as the cloud. When a medical device depends on the cloud for its essential function and safety and effectiveness, that is also part. So software bill of materials is going to be helpful for a number of different use cases, including identifying risk, because when a new vulnerability comes out inside of a piece of third-party software, a manufacturer is more quickly able to ascertain, are they at risk and is it of clinical importance? Second, it helps for the HDOs, the health delivery organizations of the world, to receive an SBOM from a manufacturer so that they too can ascertain what risks are they inheriting when they place a particular medical device in their clinical enterprise. There's much more to say on SBOM, but I'll leave that for another day. A third area that FDA is focused on is the Joint Security Plan. This was stood up by FDA through the Health Sector Coordinating Council in concert with co-leads from health delivery organizations and medical device manufacturing. And this document is a total product lifecycle reference guide on cybersecurity that is specific to medical devices. So there are many product lifecycle guides for cybersecurity available already on the internet for different markets. This one is really designed for the medical device market. And so I would encourage any manufacturer looking for how do they really integrate cybersecurity into their total product lifecycle, go read this joint security plan so you don't need to reinvent the wheel. Now, I promised I'd talk a little bit more about threat modeling. And the reason why we emphasize threat modeling so much as number one, the most fundamental thing a manufacturer can do to ensure a more successful pre-market submission is because threat models lead to better science. So, for instance, inside some of the consensus documents recognized by the FDA, such as the AAMI PIR 57 consensus standard, it talks about identification, analysis, and evaluation of potential security risks in a medical device. And as you can expect, similar to hazard analysis, the standards talk about ranking security risks as acceptable or conditionally acceptable or maybe unacceptable, depending upon various clinical aspects of the computer security risks. And the main idea behind threat modeling in good cybersecurity science is to get away from gut judgment assessments, or just statements of beliefs, and moving much more toward a verifiable security design control. And in my opinion, it's simply impossible to make scientific claims of computer security without a refutable threat model. So a few examples of threat models. We'll start with 3 threat models that I would consider inappropriate for a medical device. You might see manufacturers state something such as, well, we use an obscure programming language and so we're not likely to get hacked. That is a statement of opinion, and it's not science, nor is it refutable, and to me that does not speak for any kind of threat model. A second kind of statement that versions I've heard before are, well, we've never been attacked, therefore we never will. Again, that is not science, and that's not refutable. In fact, I would argue it's quite the opposite. If you've never been attacked, you're likely to have complacency and be more likely to have impact down the road when malware affects a medical device. A third example, a negative example of a threat model, is slightly more subtle and a little bit more difficult to unpack. I have seen instances of versions where a manufacturer might say, well, our product must be placed on a secure hospital network to be safe and effective. The problem with this statement is that computer networks are inherently hostile. They are not designed to be some kind of panacea or magic pixie dust to magically make a medical device secure. Moreover, perimeter-based controls are very 1990s, and although they are important to reduce risk, they are not an end-all be-all. A firewall is easily circumvented, and they tend not to fail so gracefully. Therefore, again, that is not science, that's not refutable, that's simply ceding responsibility. What I think is a good threat model for any medical device is to begin with a simple statement, and because many medical devices are networked, I would begin with assuming an adversary controls the computer network that the medical device connects to, even in the hospital, and having the ability for that adversary to alter, drop, and replay network packets is a very good start to assumptions to make when designing a medical device to remain safe and effective despite the anticipated and reasonably expected risks of computer security. It is a foreseeable risk. This picture over here, by the way, illustrates the problem of when you do not foresee these risks appropriately. Here is a gate on a parking lot from a long ago meme where clearly you can see it was installed, but all the parkers decided to drive through the snowy grass to get around the gate. Now, there are a number of engineering principles manufacturers can use to make medical devices safe and effective despite computer security risks. And I would tell all manufacturers to send their engineers to read this paper from 1975, way back when in the IEEE, that delineates 8 key principles, very simple principles that are extremely important to maintain the security of a system. I would just highlight 2 of these here today for the sake of time. The first one is number 4, the open design principle. It's extremely important for a manufacturer who's trying to defend their system to not depend on the ignorance of an attacker or what we call security by obscurity, where you just hope for the best. The problem is adversaries tend to increase in sophistication over time. For instance, today, the ransomware writers are extremely financially motivated and are extremely clever in their software development in order to invade a system. Instead, it's better in the open design principle to assume the adversary knows everything about your system except for instance, a small manageable cryptographic key that could be kept physically secure. A second principle I find helpful at really bringing up the strength of security in a system is something called the principle of least privilege. This is somewhat nuanced, but the idea is when you're creating a computer program or a module in some kind of software, use the least privileges necessary to complete that function. And so in computer programming, we often talk about running as a local user versus running as the system administrator or supervisor. And software tends to run much more easily when you run it as a supervisor because you never get any kind of error messages saying permission denied. But it also leads to new risk in that if that software is compromised by malware, the malware inherits all the privileges of that program. And if it's a supervisor program, it's much more likely to spread to the rest of the system. So in the last minute or so, I'll just end with medical device security is a challenging problem, and I am here in the Food and Drug Administration for the calendar year while on loan from the University of Michigan, where I'm a professor of electrical engineering and computer science. And there are 5 key areas that I'm working on involving both the strategic roadmap all the way to fostering collaborations. But I'll say that the one area for I'm really hoping to make a good dent is helping to integrate these security engineering principles into the total product market life cycle, but also helping with training and mentoring. So the FDA has recognized that cybersecurity is a key challenge, not only for post-market management of vulnerabilities and computer security incidents, but also in the pre-market reviewing. So FDA has stood up something called the Focal Point Program to bring together a lot of the FDA reviewing staff to be more consistent about cybersecurity properties during the review process. I'd be happy to take more questions, go into more detail. It sounds like we'll have a couple more minutes remaining, but I'll just end with a story and an admonition before we may have a couple moments for questions, and that is ransomware. If you pick up a newspaper anywhere today, New York Times, magazines, you will hear many stories about ransomware. In fact, there are so many stories about ransomware, it's becoming almost passé. And we are starting to let our guard down. And let me just say that ransomware has evolved to become much more sophisticated than it was even a couple years ago. Ransomware, when it breaks into a computer, will encrypt the local storage systems and hold it ransom until you pay usually through some kind of semi-anonymous Bitcoin payment system to get the cryptographic key back in order to get your data. That was the sort of version 1.0 of ransomware a few years ago. Today, there are teams of ransomware writers that will actually scout out a target ahead of time, break into an enterprise or a medical device company. They will silently disable all your backup systems, and then sometime later, when you're least suspecting, they will release their ransomware. And when the system administrator goes and thinks, well, we have a plan, we have backups, they go to look for their backups, and guess what? They don't exist. This is the new world of ransomware, and this is why you're hearing about outages of hospitals and medical device manufacturers being unavailable to deliver care for days and sometimes weeks because of computer security incidents. So we at FDA are working hard on the standards and guidance to get out in front of this problem. We've already released these guidance documents, and we'd like to see these systems used to prevent the problems. Thank you.
So thank you very much for your great but scary talk. And I guess we don't really have time for questions. We'll we will move on to
All
our
right.
next
Thank you
speaker,
for being
which is
here.
Larry Lee. But thank you. Yeah. Hang on.
Hi, good afternoon, everyone. My name is Larry Lee, and I'm the Deputy Director of Science in the Office of Pharmaceutical Quality. And today I'm going to highlight the CEDAR effort in facilitating the adoption of advanced manufacturing. It is important to first talk about the importance of pharmaceutical quality as mentioned by the first speaker. In general, a quality product means that it consistently meets the expectations of the user. Just no difference. To understand the importance of pharmaceutical quality, it is necessary to relate pharmaceutical quality to patients' perspective. Specifically, patients expect safe and effective medicine. With every dose they take, pharmaceutical quality is consistently meeting standards that ensure every dose is safe and effective, free of contamination and defects. It is what gives our patients confidence in their next dose of medicine. And advanced manufacturing include innovative manufacturing methods, new dosage forms, or delivery systems, as well as advanced analytical tools. From our perspective, we believe that advanced manufacturing is key to advancing pharmaceutical quality. For example, advanced manufacturing will further enhance our ability to deliver quality drug products over the life cycle. It also will enable flexible and agile manufacture of high-quality drugs, as described, as also mentioned by the first speaker, without extensive regulatory quality oversight. Furthermore, advanced manufacturing support new clinical development to meet unmet patient needs, shorter product development or manufacturing development timeframes, and improve our capability in response to a pandemic such as the COVID-19 situation we are facing right now. One noticeable example of advanced manufacturing is continuous manufacturing. Let me summarize some key aspects of our approaches to facilitate adoption of advanced manufacturing technologies. We have a vigorous intramural and extramural research program to improve our readiness in evaluating new technologies based on the science and risk-based approach. We established the so-called Emerging Technology Program for sponsors to get early feedback from the agency regarding their proposed technology. And I will describe this program more in the next few slides. As we gain sufficient knowledge and experience on new technologies, we will facilitate development of relevant regulatory guidance documents to guide industry for product and technology development. To facilitate the adoption of advanced manufacturing, we established the Emerging Technology Program. The vision of this program is to provide a centralized, focused, collaborative platform within the FDA, as well as between the FDA and industry, so we can work together to move new promising technology forward. This program is led by a cross-functional emerging technology team, the so-called ETT, with representation from all relevant FDA quality review and inspection programs, including the Office of Pharmaceutical Quality, Office of Compliance, and Office of Regulatory Affairs. To clarify the mission, scope, and process of this important program, we have issued the FDA guidance in the area of Emerging Technology Program. This program has several objectives and functions. ETT serves as a centralized location for all external inquiries on novel manufacturing technologies, by providing an opportunity for the sponsors to engage in early scientific or regulatory discussions with ETT and possibly relevant quality assessors within our office. ETT also ensures consistency, continuity, and predictability in review and inspection based on science. Furthermore, ETT helps identify and evaluate potential roadblocks related to existing guidance, policy, or practice, as well as establish quality standards and policy for new technologies as needed. ETT facilitates knowledge transfer to relevant FDA quality review and inspection programs. When the emerging technology becomes mature, ETT also engage with international regulatory agencies to share their learning and approach as part of the effort to promote global harmonization on standards in the area of emerging technologies. This slide shows some examples of a novel technology we encounter under the Emerging Technology Program. As you can see the list here, we covered innovative technologies for small molecules and biological molecules, as well as for multiple products. And based on the experience we have gained in advanced manufacturing, such as continuous manufacturing, the Emerging Technology Program, we have published a guidance on continuous manufacturing in 2019 with focus on solid oral dosage forms. Recognizing that a broad adoption of this type of a new technology will require a global effort, we supported and actively participated in the development of the ICH Q13 on continuous manufacturing of drug substances and drug products. The objective of this guideline, international guideline, is to capture and harmonize the key technical and regulatory considerations specific to continuous manufacturing, while it still allows manufacturers to explore flexible and innovative approaches to develop and implement continuous manufacturing for the manufacturing of small and large molecules for the new and existing products. The Q13, the expert working group, expects to have a step 1 sign-off at the end of this month. We have started Advanced Manufacturing Regulatory Framework initiative to modernize our regulatory framework and policy so we can facilitate the implementation of technology like distributed manufacturing, as mentioned by the first speaker, and provides flexibility for additional novel technologies. For the purpose of our initiative, we consider that distributed manufacturing is a decentralized manufacturing platform that is coordinated by one management, is mobile, and can be deployed to multiple locations. Distributed manufacturing typically comprises of prefabricated, mobile, modular manufacturing and testing units. Oversight is established by centralized QA. Execution of manufacturing occurs where the unit is deployed. And the potential benefits of distributed manufacturing include better microbial control because it is self-contained, reduced cross-contamination, and is cost-effective alternative to traditional clean rooms. Supply chain flexibility because of its portability, because you can put the machine anywhere you want. And product development speed because it really facilitates quick tech transfers because you can move this machine around. Several use case scenarios for mobile manufacturing platform, the so-called PARTS or Pharmacy and Demand, have been discussed under the Emerging Technology Program. This includes implementing PARTS or the so-called Pharmacy on Demand within the existing facility, relocating this type of a technology or manufacturing platform between existing facilities, replacing this type of technology, replicating this type of technology to increase capability, and rapid response to public health by moving this mobile manufacturing platform closer to the point of care or hospitals. One key challenge is that mobile manufacturing platforms can move to multiple locations, which will require innovative thinking on pharmaceutical quality system as well as the regulatory frameworks we have currently. We are continuing our efforts to develop a sensible policy, solution, and regulatory framework to the pain points that we have identified in order to implement this type of a technology, as well as going to engage relevant stakeholders for their input. We have a rigorous science and research program to support the emerging technology program. We have been applying our in-house laboratory and computational capability to study various new technologies, as well as collaborating with external stakeholders, such as universities like Rutgers, Purdue, and MIT, in research on advanced manufacturing. The knowledge generated in this research activity, together with the information provided by the sponsor, will facilitate more effective quality assessment of this technology. And I will highlight two examples in the next two slides. The adoption of continuous manufacturing has been a driving force for the growing utilization of modeling and simulation for risk assessment, process design, and control strategy. And the amount of modeling and simulation data submitted in the drug application to support CM continues to grow. We established a computational group in our laboratory to develop and use modeling simulation tools to aid regulatory assessment of advanced manufacturing. This group supported the Emerging Technology Program to evaluate model-based approach in drug application, as well as a policy and guidance development such as ICHQ-13. This approach includes the digital twins, as well as using residence time distribution model that are used to predict impact of material feeding disturbances on the blend and contact uniformity in the real time, as illustrated in the figure on the right. Another example is the MAM multi-attribute method, which is LC-MS-based method that has been proposed to Emerging Technology Program as a replacement for conventional QC method for release testing of therapeutic proteins. The figure shows high-level overview of this method. Digest protein analyze the digest proteins with LC-MS and database searching to fully characterize the product. And based on our research and understanding of these applications, we have published a prospective paper in 2019 highlighting 4 major areas to consider for QC quality control implementation, including risk assessment, method validation, new peak detection, and comparison with conventional method. We have developed internal MAM capability using monoclonal antibody as a model protein. Our MAM research has supported the evaluation of multiple drug application using MAM. This application involves products at different stage of product development, and for one sponsor, we agreed to sunset strategy for the conventional methods to be eventually replaced by this new analytical method. Moving forward, we will focus on 4 areas to facilitate the adoption of advanced manufacturing. First, we have started program improvement for the Emerging Technology Program to meet increasing workload in the near future. Second, we continue our initiative on advanced manufacturing regulatory framework to make sure that our regulation and our policy is not the barrier for this type of a new technology. Third, we are building research manufacturing pilot plant facility to increase our capability to conduct research and training in advanced manufacturing areas. And finally, we will synergize our advanced manufacturing effort with other initiatives such as quality, maturity management, and ISHQ 12 to further improve and streamline the effectiveness and efficiency of regulatory oversight of the product manufactured by this type of a new technology. And with this, I conclude my presentation and thank you for your attention.
Thank you very much, Larry. That's very, very interesting. And I can't really think of any questions. So why don't we move on to our last speaker? And I'm sorry, I'm probably going to say your first name wrong, Pankaj Mandal. And I apologize if I didn't say that. But thank you for presenting today.
Hello, good afternoon, everyone. My name is Pankaj Mandal, and I'm a senior staff fellow principal investigator in Center for Biologics Evaluation and Research. And today I'm going to talk about understanding ex vivo manufacturing of hematopoietic stem cell-based therapeutics. So here is the outline of my talk, introduction followed by what are the challenges and limitations of HSC-based therapeutics and how we can understand the advanced manufacturing of cellular therapeutics, and briefly I will outline the ongoing research activity in my lab. So here are the learning objectives. By end of this talk, we will know about hematopoietic stem cells and hematopoietic cell-based therapeutics, and what are the manufacturing challenges associated with cell therapy products. So hematopoietic stem cells and in general hematopoietic systems are at the forefront of cell-based therapy and regenerative medicine. And as you see here, both allogeneic hematopoietic stem cells transplantation and autologous hematopoietic stem cell transplantation have been used for treating various hematological disorders and hematological malignancies. And as shown in the bottom of the graph, the trend is increasing, and so far over 1 million transplantation has been performed worldwide. So just to give you a brief overview of hematopoietic system is generated and hematopoiesis is maintained by hematopoietic stem cells, which is at the top of the hierarchy of the hematopoietic tissue. And through the process of differentiation, it gives rise to multipotent progenitor cells followed by common myeloid progenitors and common lymphoid progenitors. And that ultimately, through a multi-step differentiation process, give rise to effector cells of blood system, including red blood cells, macrophages, neutrophils, basophils, eosinophils, and B and T lymphoid cells and NK cells and others. So why hematopoietic system is at the forefront of regenerative medicine? It's because of easy accessibility to the tissue, and I'm sorry because slides are not looking good. One can easily obtain peripheral blood that can be manipulated ex vivo and modified with gene therapy or genome editing reagents, and those can be transplanted back for therapeutic benefit into the patients. What we ideally want to do is expand these cells ex vivo in large numbers so that a large number of patients can be treated with a single batch of the drug product. So, as I said, hematopoietic system are on top of the hematopoietic cell tissue hierarchy. Any interventions that is made in hematopoietic stem cells will translate into all the downstream progeny derived from HSC through differentiation. Not only that, by the process of self-renewal, hematopoietic stem cells maintain itself throughout life, so that any intervention which is made in hematopoietic stem cell will have a lifelong effect and can provide a long-term cure. So here are a list of diseases of hematopoietic system affecting such as sickle cell disease or beta thalassemia, affecting RBCs, SCID mutations, ADA and IL-2 receptor gamma mutation affecting the lymphoid compartment, and so on and so forth. So as you can see, intervention in hematopoietic system allows us to provide a cure for many of the hematological disorders and diseases that we are currently in the process of developing. So, as I said, hematopoietic stem cells are regenerative unit of blood system. Both autologous and allogeneic HSC bone marrow transplantation is routinely done. And with the advent of gene therapy and genome editing, there is a broader applicability of hematopoietic stem cell-based therapeutics. So, however, since hematopoietic stem cells are hard to obtain, there are challenges and limitations associated with such therapeutics, and I will go through in a moment. So bone marrow transplantation, though it is a routinely performed clinical procedure, over 1 million transplantation has been performed worldwide, yet it is an underutilized procedure to treat the blood disorders. Because of the associated risk, it is a high-risk procedure with a 1-year survival of up to 70% in HLA-matched donor, approximately 55% in unrelated donors, and further compounded by relapse, opportunistic infection, and graft versus host diseases. The number of HSC transplanted is a strong predicator of transplantation success, and as we grow these cells in ex vivo environment, they tend to lose their stemness, and therefore it is challenging to expand it in enough number. And HSCs are exceedingly rare in the bone marrow, and method to maintain these cells ex vivo hasn't been developed yet, though the current progress is quite encouraging. So when CRISPR came in picture, we decided to study the efficiency of CRISPR-Cas9 editing in hematopoietic compartment, and this is a published story a long time back, and we showed that the CRISPR editing in hematopoietic stem and progenitor cells is highly efficient. The edited cells maintain their stemness both in vitro studies in colony-forming assays as well as in transplantation study in vivo. The edited cells persist as shown here, the persistence of the deleted or edited cells, and we did a thorough off-target analysis and found that these cells are having less off-target effect compared to cell lines, and these are tolerable. So since then, the field has evolved tremendously and have grown exponentially, and now we have many flavors of genome editing tools, including Cas9, dCas9, based derivatives and other Cas proteins. We can achieve a therapeutic meaningful genome editing with minimal off-target effects, and the potency, safety, and durability of such a treatment is yet to be established, but earlier clinical indications suggested that these are safe and durable. So here is an outline of treatment pipeline. It begins with the HSC isolation, mobilization, and peripheral blood apheresis, followed by HSC enrichment, ex vivo manipulation with gene therapy genome editing reagent, ex vivo expansion of the cells, followed by harvest and formulation, and then ultimately it goes into the patient. So this step of mobilization and peripheral blood apheresis remains a standard clinical procedure, and same the infusion into the patient. The most challenging part is manipulating these cells ex vivo with high efficiency and robust expansion in vitro conditions while maintaining their stemness are the bottleneck for developing such therapeutics for broader applicability. So though we have efficient therapeutic meaningful genome editing in place, and safety of these cells are quite well established, and what we are struggling or is a bottleneck is expansion of a therapeutic dose that can be used. So efficient genome editing together with robust ex vivo expansion will be transformative for transplantation medicine. Why it is important for studying advanced manufacturing, as Dr. Lee alluded, how the advanced manufacturing is assisting in drug development. It's very important to study advanced manufacturing of cellular therapies. As alluded in this report on advanced manufacturing subcommittee, a significant challenge of development of regenerative medicine products and therapies in the field is still transitioning from an academic enterprises to an industry-based commercial enterprise. And because of manufacturing cell therapy products are in itself a very complex and challenging process. So it is significantly more complex, and therefore it is important to study the advanced manufacturing to have a better control over manufacturing products and processes, to define the critical quality attributes and critical and release criteria, and to develop the regulatory guidelines for this product class. So here are the two models. One is centralized and another one is near patient manufacturing model, with both having their own advantages and disadvantages. I will not go into the detail. And there are currently, for autologous hematopoietic stem cell based therapies, there are many closed and single-use platform which can manufacture the HSC-based therapies for individual patients, but the challenge remains to study large-scale manufacturing of this product class. So in 2019, I started my lab to study advanced manufacturing and evaluation of genome-edited HSC-based therapeutics with the aim to optimize large-scale manufacturing of HSC, to develop novel reagents and approaches for HSC derivation and expansion, and to evaluate the potency and safety of genome-edited HSC-based therapeutics. So the tools that we are using was generated in my postdoc, that we identified HSC restricted gene, and one example shown here is FGD5, which is highly expressed in hematopoietic stem cells and not in any other progeny. And we designed a reporter construct and knocked into the locus to generate a reporter strain of mice, which marks hematopoietic stem cells, which is characterized by lineage negative, c-KIT positive, SCA positive, CD48 negative, and 150 positive. And we validated these that reporter positive cells retains HSC activity, and reporter negative cells is not able to maintain reconstitution in long-term assays. And we are using this tool to study advanced manufacturing of HSC-based therapeutics. So our aim is to optimize large-scale manufacturing of HSC-based product, developing novel approaches for HSC derivation and expansion, and evaluate the potency and safety of the genome-edited HSCs, and development of reference reagent and regulatory guidelines. And here are the people in my lab who are putting tremendous effort in understanding advanced manufacturing, and this is my contact information, and I will be happy to take any questions. Thank you for your time and attention.
Thank you very much, Dr. Mandel. And I don't know if we have any questions here, but if people have questions, you have his contact information. So at this time, I will pop back on, I think, and here I am. Thank you to all the speakers for a great presentations, and thank you for the audience, and remind you that this is the end of day one of the Science Forum, but we have more exciting science tomorrow beginning at 8:55 a.m. So please come back on day two and see more of our science presentations. Please go see the virtual poster exhibition that we have at fda.gov. And most importantly, thank you to Michael Kay for making all this possible with all his hard work and keeping us in mind. So thank you, everyone. Have a good evening, and we'll see you all tomorrow. Bye.
Thank you.